All right, now that we're done with experiment one, let me briefly give you this video about experiment two. All the nitty gritty, all the hard stuff you learned about in experiment one, like how to do the calculations, where to get your data, what the data means, all that stuff. So for this video, I'm strictly going to basically just show you how to do it, how to do experiment two. And again, you know how to do most of it because most of it hasn't changed since experiment one. So mostly what I'm going to focus on are the differences between experiment one and experiment two. So here we go for experiment two. And I'll remind you now we're talking about gene flow, right? So now we're going to see how this population of fish changes over time. You know, the orange fish and the spotted fish and the white fish. We're going to see how the percentages of those fish change over time. But instead of like last time where it was genetic drift, where it was all by chance, now we're talking about actually gene flow. And before we get to it, as the next set of instructions says right here, first of all, we need to reset where we started. So let's go back here. And you can see this is where we ended experiment one, and we're done with experiment one, and we don't need this data anymore. We don't need those parameters anymore. So we refresh this, and then we are back where we started. So, okay, there we go, back where we started. Now let's talk about experiment two very briefly. This time, because you were simulating gene flow, you're going to need to change the parameter that's actually for gene flow. And you're going to find that under evolutionary parameters. And then under evolutionary parameters, you're going to find migration. And then under migration, you're going to find migration rate. And that's the part we're going to switch. And obviously, I'm reading this now, but I'm going to show it to you in the video and actually show you how to do it. What we're going to need to do for the part that says migration rate, we're going to click on the up button. And again, I'll show you this in action. I'll show it to you, me doing it on the website. And once you hit that button once, it should then read 0.1. And like I keep saying, I don't want you to just follow these directions blindly, just to get the information so you can get the lab done. Right? All right, first click this, then click that, then write this down. That's not what it's about, right? I want you to understand what it means when you click this and what it means when you click that. So when you click this, the migration rate button, and you click it up, and it now reads 0.1%. What this means is that for every generation, and remember, we're doing 100 generations, for every generation, the equivalent of 10% of the population will immigrate from another population into this population. And again, I'm going to show you all this when I bring it back to the other screen. So that's what we'll do with that one, with that button. The other button is the migration R, big R allele button, and that should already be set to 1, and that's what we need it to be. And what does that mean that it's set to 1? Again, I want you to know what the numbers mean, I don't want you just plugging numbers into a machine and finding the results. This means that for every fish that immigrates into the population, and we already said for every generation, 10% of the population will immigrate in, right? So for every one of those fish that comes in, the number of the R alleles in the gene pool is going to increase by 1. So now that I've said all that, and I understand that might be a little bit confusing because all you see is the words, and I've said it very quickly, and you haven't seen it in action. Let's see it in action. So here's what you need to do. Again, you've already done the refresh, right? So we're starting new. We've erased everything from experiment one. We're doing experiment two. So we're going to hit this here, run experiments link. And remember, we're going to need to pause the experiment right away because we need to set our parameters. So be ready to pause it right after you hit this run experiments link. So here we go. Hit that and pause. All right. I did it as quick as I could. Again, just like in experiment one, all this is set where it needs to be. You don't need to change it. Here's the evolutionary parameter that we're working with. We're working with migration because, again, it's gene flow. So we are going to simulate fish coming in. So the migration rate is up by 0.1, meaning for every generation, again, 10% of the number of fish that are already in the pond right here, whatever 10% of that is, that's going to be coming into the pond from some other place, right? It's immigrating in. And of course, given the fact that we have two different alleles, right? We've got big R, we have little r, right? The dominant or the recessive allele. Given that fact, we would wonder what is coming in. So how much of these coming in are going to be the big R allele? and it's going to be 1, right? So basically what we're saying is that every single fish that comes in here is going to be big R, big R. That's all that means. And again, the mutation rate is set where it needs to be 0 to 0, so leave that alone. The genotype, relative fitness, which is the thing that represents or simulates natural selection, that's exactly where it needs to be, 1, 1, 1, meaning everything is equal. Same thing here with the assortative mating, which simulates sexual selection, and it's set to 0. So again, everything is set to where we are only simulating gene flow through migration. Now remember, before we hit the play button, we need to collect our data because, let's go back here, the whole purpose of this experiment is to test your hypothesis. And to be able to test your hypothesis, excuse me, let's assume that wasn't there, sorry. To test the hypothesis that you already gave for experiment 2, you need to run the experiment, you need to collect data, and then you need to analyze the data. And that's what this table is for, as a reminder. So the first thing we need to do is to collect the data from here, right? We need the initial data. So we've already set our parameters. So we need to go here to data, right? And this is our initial data. So all this information right here, just like it was in experiment 1, is going to be used to fill in this right here. And then once you've filled this part of the table in from all the initial data right here, just like the last experiment, you're going to hit play. You're going to wait for it to hit 100 generations. Again, when you hit play, it defaults to the slowest speed, so you can speed it up. I'm going to speed it all the way up to 16, which is the fastest. And again, we're trying to hit 100. As you're watching it, you can look at the data and see how it's changing. You can see how the population size is fluctuating, but still, for the most part, over a longer distance of time or a longer time period, it's staying the same. You can see here fish coming in and out. You can see how they're all orange, right? Because again, they're all big R, big R. You can see here where the big R is now the majority and the rest of them aren't. So look, we came close to 100, so I paused it. Just like in experiment number 1, you're shooting for 100, but if you miss it by a few, that's okay. So I happen to hit 103, so I'm going to come over here and say after 103 generations as opposed to after 100 generations. And just like in experiment 1, if you feel like it, you can just keep it going, right? You don't have to stop at 100. You can go as long as you want, as long as you hit at least 100, right? You're trying to hit at least 100 generations. And if you feel like doing more, that's fine. Just make sure that you update this portion of your table. So anyway, we are now done running that experiment, and now we have all the data we need. So we're going to take all this data right here at the top left from this table, and we're going to put it in right here. Right? We're going to use all that data to fill this data in. And at that point, you are done running the experiment. You are done collecting data. So from that point, you're going to use the data you have to calculate the difference between initial and after. Right? And then once you have that data, then you will then be enabled to calculate the percentage change from initial to after 100 generations. And again, I'm going through this quickly because if you want to know how to do these calculations, the difference calculations or the percent change calculations, I showed you in the previous video, and also again, it's written right up here. So that's how you do experiment 2. That is the end of this video. And experiment 3's video will be just like this, except it's going to be just the minute differences between the the experiments.